when we first got the people together, they told me that they had never, ever been together in the same room at the same time. These are Chinese, the top Chinese surgeons don't work well with each other. They're very competitive, and this is the first time ever that they have joined forces to work together. And it's very interesting. The reason why they tell me they do this is because they now understand the only way they can get rid of all the non-effective therapies in China is to join forces, to test therapies, and if therapies doesn't work, they have the data in which to say, don't use that therapy. And they understand that this is a very major problem in China, and they have to do this by collaboration. It gives me really warm feelings inside when I go to one of our meetings. We've been meeting now consistently since 2004, and people are more excited about the China Spinal Cord Injury Network than I can ever imagine them to be. Everybody here knows about clinical trials, phase 1, phase 2, phase 3, phase 4, so I'm not going to go through them. From the very beginning, we decided that the therapies that we have to orient our clinical trials towards is combination therapies. And so there are essentially three major problems that have to be solved in regeneration spinal cord. And one is to bridge the injury site, the second one is to provide sustained growth factor support, and the third one is to block growth inhibitors. So we searched for possible feasible cell transplants, and the key word is feasible. We looked for methods of sustaining growth factor support, and thirdly, we evaluated treatments to block growth inhibitors in the spinal cord, and that's what we wanted to do. We wanted to aim to get these things into trial. So everybody has seen lists like this. These are the different potential cell sources. After looking very carefully at all of these, in fact, I interviewed Woo-Suk Wong on two occasions. I've talked to many groups in terms of cell sources. We came and decided to focus our effort on umbilical cord blood. And the reason we did that was as follows. First, a number of independent laboratories have reported beneficial effects of umbilical cord blood transplanted into spinal cord. Now, the first two studies are sort of not credible. One is by Saporta, 2003, and one is by Zhao in China. And basically, they use human UCB cells, and one used a clip-compressed cord, the other used hemisected spinal cords. And I just didn't find these studies all that believable. However, all these three studies were done with the contusion model. They're done by people I trained. And even more important, I think the results really fit with what we have seen in our lab as well. So basically, Koo in Seoul, Korea, found that human UCB cells don't survive in the spinal cord very long. And by the way, none of these used cyclosporin. They only survive for about 3 weeks, and only when you give BDNF along with human UCB cells did one see improved walking, and then if you do either one alone, you don't get improved recovery. And Nishio in Chiba University did CD34 enriched cells, and they found that this improved walking. Dasari published 3 papers. He's from Illinois. They're from Illinois, and they did human UCB cells into contused spinal cords and found improved walking. We were very much influenced by the fact that there are human umbilical cord blood banks that are available that would can provide HLA-matched cells to us, and that this is a long-established GMP source of cells. And this is what Hans talked about. GMP was a tremendous concern to us, and we didn't want to spend 5 years developing a GMP source of cells that would be needed. So we decided to go for this particular GMP source of cells. Now, the alternative to this is bone marrow stem cell transplants, either stromal cells. But to do that, we would have had to develop GMP labs in each one of these clinical trial centers, and we could not afford to do that. So this actually was not defined by as much science as much as by feasibility and the presence of some data indicating potential benefit. And then in 2004, my colleagues at Hong Kong University published this paper in Journal of Neurotrauma, which is really very interesting. They found out that chondroitinase and lithium together stimulated regeneration of rat spinal cords, but each one alone did not. So in 2005, this paper was published in November of 2004. 2005, came back to the lab. I said, listen, let's put some lithium onto cells to see what it does. Because I had always thought lithium was just for manic depression. Who knows? Well, to our enormous surprise, what we found was it causes cells to proliferate. And the first cells that we tried it on were rat neonatal blood cells, and it caused the mononuclear cells to proliferate like crazy. By the way, it also causes embryonic stem cells to proliferate. In fact, lithium is part of a standard non-serum based media. If you look at the contents of that, there's lithium inside the media. And we also found out that particularly in blood cells, and particularly in mononuclear cells from human umbilical cord blood and from rat, we found that it stimulates the cells to produce a huge amount of neurotrophins. We have some unpublished data that now have worked out the mechanism by which this occurs, and I'll just talk about this briefly. It inhibits an enzyme called glycogen synthetase kinase beta 3, and GSK beta 3 normally phosphorylates a nuclear factor called NFAT, nuclear factor of activated T cells, and NFAT, when it's phosphorylated, cannot get into the tissue, into the nucleus. NFAT is the target of calcineurin, which is the enzyme that cyclosporin inhibits. And so when NFAT enters the nucleus, it turns on all these genes for both proliferation as well as neurotrophin production. And we see a 100-fold or greater increase in secretion of several key neurotrophins when we give lithium to human umbilical cord blood cells. One of them is GDNF. Second one is NT3. Third one is NGF. And the fourth one is LIF, leukemia inhibiting factor. And these are all factors that have been reported to stimulate regeneration in the spinal cord. So we're very happy to see that. And we thought that lithium would be a very good drug to do an initial study on to see whether or not we can provide sustained growth factor support. So if you remember, the first of the combination was to provide cells, the second one to stimulate growth factor support, and now the third one is the axonal growth inhibitors. As many of you know, there are a number of growth inhibitors have been identified. These include the NOGO family and MAG and other myelin-based proteins. There's CSPG, and there are a number of molecules that play a role during development. What is very nice is that there are drugs that have now been shown to directly block these, some of these. And there are now 2 NOGO-A receptors that are in clinical trial, or soon to be in clinical trial. This is Novartis's drug, anti-NOGO antibody. It's based on the IN-1 of Martin Schwab, and they're now finishing their phase 1/2 trial in Europe. There's a NOGO receptor protein that's made by Biogen, and this is a very, very nice molecule, protein that binds onto NOGO-A very tightly. There's a NOGO66, which is a Strip Matter's original NOGO receptor blocker, and then there's Cethrin, which is a recombinant C3 toxin, which inhibits RhoA. Cethrin has gone through a phase 1 trial, and I think in 30-something patients, has actually positive results, a 1/3 conversion rate from Asia A to Asia C. A company, the original company that made this with Lisa McCarricker now has been bought up by Alcerez. Alcerez used to be Boston Life Sciences, so they're going forward with this thing. I am a little worried because I've heard that Alcerez is running out of money. But nevertheless, it's a very, very good candidate. Biogen has lingual receptor blockers that are very interesting. There's also the possibility of blocking P75 receptors. And finally, there's chondroitinase and a number of CSPG enzymes. So we would very much like to